So we were talking about Chargaff's rule. So this Chargaff, so we have already done this. What is nucleic acid? So this nucleic acid, you know, are bonded by phosphodiester bond, and this phosphodiester bond is between 3' to 5' carbon, 3' to 5', and the nucleic acid has free 5' this phosphate and free 3' tail. This is called tail. So this is tail end and this is head end. This is head end. Now head to tail. So this is we will, that is why we label this any DNA RNA in this sequence 5 to 3'. Is 5 to 3'. Direction of polymerization we take is from 5' to 3'. So, and this is Chargaff based on this his finding, and I've already told you this. He found that purine is equal to pyrimidine, and this purine, adenosine is equal to thymine, and guanine is equal to cytosine. So based on his finding only, adenosine is finding only, double standard helical model was proposed by these two scientists. There are one more scientist, Franklin. So Watson and Crick discovered this double standard model based on his finding of this base pairing. His finding on base pairing, purine pairs base with pyrimidine, adenine with thymine, and guanine with cytosine. But he was unable to make connection. And actually, when they got Nobel Prize, these two persons got Nobel Prize. When they got Nobel Prize, and he actually opposed. This man, Chargaff, opposed for that Nobel Prize. He thought that he should also share this Nobel Prize with them, but he was not included. So remember this, this whole finding, the double standard helical model was based on this Chargaff principle. So how does this adenine is equal to thymine and this guanine is equal to cytosine? If you don't understand, please. So you can see this is adenine and this is thymine. So this adenine, as you see, adenine has got amine group here, and this thymine has got this 2O group. This adenine will make bond with this. This adenine will make bond, do 2 bond with, this is hydrogen bond, 2 hydrogen bond with thymine. So adenine makes 2 hydrogen bond with thymine, and you can see how does it make bond. It makes bond with this amino group. I have already discussed this amino group. There was one amino group. This is amino group. You can see thymine has got one amino group, and this amino group has hydrogen, and this with this hydrogen, this oxo group will make one bond. With this thymine, with this hydrogen, this nitrogen makes this hydrogen bond. So 2 hydrogen bond is found between this thymine and guanine. Now let's see this. So this let's talk about this guanine and cytosine. So guanine.
Sorry,
yes.
Just to confirm what you said, 2 hydrogen bonds are found between adenine and thymine, right?
Yes, adenine and thymine. Yes, great. And then bond is formed between guanine and cytosine. You can see here. So guanine here, and guanine has this structure. You can see this structure. One oxo group, and here in between 2 nitrogen, there is nitrogen, ammonia group. And this ammonia group will form bond with oxo group of this cytosine here. And this oxo group will form this hydrogen bond with this amine group. And here you will find this hydrogen bond will form bond with this nitrogen of this cytosine. So you will find there is 3 hydrogen bond between guanine and cytosine. So can you see 3 hydrogen bond here? Can you see 3 hydrogen bond here? Can you see 2 hydrogen bond here? So 2 hydrogen bond formation here and 3 hydrogen bond formation here. So based on this, because this thymine and adenine makes hydrogen bond, this guanine and cytosine makes hydrogen bond. So the hydrogen bond formation make DNA double stranded. So DNA is single stranded and this is single stranded. So because of this presence of this hydrogen bonding between two bases like this, so there is formation of hydrogen bonding. That is why this DNA is double stranded. And this double stranded DNA, you can see this is a backbone. You can compare this whole DNA structure like a stair. Like a stair. In that stair, this is backbone. In that backbone, you can compare this as a stair. Steps. These are the steps in the stair. These are steps in the stair. So these steps are very horizontal. These steps are very horizontal. Okay? These steps are very horizontal. Very horizontal. It is not like this. It is not like this. It is very horizontal. Steps are very horizontal. And these steps are made by bases. Okay? These steps are made by bases. And this backbone, this is made by this backbone of this, that what you consider this side support is made by, as you can see, sugar and phosphate. Sugar and phosphate will make this backbone. So this is how the DNA helix is formed. So just understand, this is two part of backbone, and in that backbone this base is straight wise. It is not like this. It is this, the shape is like this. So what will happen is this base, in that sense, how DNA structure is stabilized is this base will form a stacking on other base. Other base will form a stacking on other base. Other base will form a stacking on other base. So there is a hydrophobic interaction between these two stacking. Though I have shown here a bit in distance, but they are very closely approximated, and they are like this. So this close stacking of the bases, and this is close stacking is because of hydrophobic interaction. This close stacking is based on hydrophobic interaction. Now let's talk about this DNA structure. So DNA, it is like a stair, and this stair will rotate. So DNA helical model is like this. Why? Because this DNA adjusts itself in environment of water, because our body is made of water. So what happens is these bases are hydrophobic. So this bases, and but this backbone, if you can see this backbone here, these backbones you can see hydrophilic regions are there. So phosphate is hydrophilic, this sugar OH is also hydrophilic. These are hydrophilic. So this hydrophilic part are in contact with water, but these bases, they are hydrophobic. That is why they will try to hide in the water. That is why they will rotate, and this are away from, try to hide from the water, and then this will provide, this two will provide the core, though they will try to limit this hydrophobic region in contact with water, and that is why this DNA rotates. It is in order to give it stability. This gives it stability, because DNA, if you see, it is made by endergonic process. So anything that is made by endergonic process that require energy, for example, wood, when you fire, it will convert into carbon dioxide. So it is not that thermodynamically that stable, but it when it rotates, it makes it stable. So this is how this DNA attains double-stranded twisted model, right-handed double-stranded model, double-stranded helical, double-stranded because of this hydrogen bonding, and this right-handed twist is because of interaction of water and hydrophobic interaction. So hydrophilic and hydrophobic interaction make it twisted. So this is the result of this DNA structure. Now you can see twisted right-handed, you can see right-handed double helix. So DNA is called right-handed double helix. This is right-handed double helix. Now we will see the parameter of that right-handed. What is the length and what is the breadth of that right-handed DNA helix? So this DNA helix are anti-parallel. What does it mean? One strand, as you have seen, this one strand will go from 5 prime to 3 prime, and other strand will go from here like this, in this direction, 5 prime to 3 prime. So one will go this direction, other will go this direction. So this 5 prime to 3 prime and 3 prime to 5 prime. So this is double-stranded helical model of DNA. It was proposed in 1953 and got Nobel Prize in 1962. This you can imagine like this is right-handed. If you do your hand like this, so this folding is right-handed. And if you do like this, it is left-handed. So right-handed, this is DNA helix, and this is left-handed DNA helix. You can see here. So this is DNA helix. This is right-handed DNA helix. You can see it like this. You can compare this with this. So this is right-handed double-stranded helix. Now double-stranded helix, if you see the double-stranded helix, so this helix, width of this helix is 20 nanometer. This is important, 20 nanometer or 2 nanometer. So width is 20 nanometer or 2 nanometer. And if you see the space between these two bases, distance between two bases, so this is width, and you can consider this as length. If you consider this width between these two bases, it is 44 angstrom. So length is 34 angstrom, and that means 3.4 nanometer, and this width is 20 angstrom or 2 nanometer. So this is easy to understand. Always generally length is more than breadth. That way you can understand this. And in that length also there is one parameter, one pitch, one complete fold. When DNA is folded completely, so this DNA is complete fold of this DNA has about 10 bases. About 10 bases. And in this 10 bases, if you multiply this 10 with 3.4, what it becomes? 34 nanometer. So length of this one pitch is 34 nanometer. Length of this two bases, between these two bases, is 3.4 nanometer. Understand this. So these are the width and height of this DNA, length and width of this DNA. Now DNA, when you fold this, when DNA is, just consider DNA as stair only, not for unfolded DNA. So this is DNA. So when DNA, you place DNA like that, so one side of this stair is major group, other side of this stair is minor group. And when it folds like this. So this major group, you can see major group, the width of major group is about, just remember, about 1 nanometer. So remember in a rough way, then you will remember. And this width, so width of this minor group is 1 nanometer and width of this major group is 2 nanometer. So there is significance of major group and minor group. We'll talk about that in later. So you can see the major group. So major group is, if you consider this DNA like this paper, if you consider DNA like this paper, like this paper, if you consider this DNA like this paper, So you can imagine this is a backbone. Here is also backbone. So it is not yet right-handed. It is not twisted. So I have considered DNA. I have straightened the DNA. When you do like this, so what will happen? This part, the DNA actually has this shape. So base has this groove. So this is backbone and this is backbone. So this is like this. DNA will be like this. Base will be like this. You can see here. When there is, this is like here sugar is attached. Here also sugar is attached. So what will happen is one side of this base, one side of this base has a acute angle. Other side of this base will have obtuse angle. You can see this because this bond is at 9th position and 1 position. That is why acute and obtuse angle. And this acute and obtuse, because of this acute and obtuse angle, what is formed is this like this. So it is formed like this. So what will happen is, so this becomes major group and this becomes minor group. So you can see acute type of group becomes minor group and this becomes major group. And this major group has width, as I have told you, 2 nanometer. And that means 20 angstrom, about 20 angstrom, 22 angstrom. And minor group has width only 1 nanometer. So this, you can, all this major group and minor group has very important implication. You can say this is minor group and opposite is major group. This is very important implication. So this major group, at major group and minor group only protein binds. Generally, because DNA synthesis is also done by DNA polymerase, and this is also a protein. And DNA expression and repression, it is also done by DNA, this protein, and protein has to bind to DNA. Where does this protein bind to DNA? Protein will bind to DNA in this major group generally. Generally, protein will bind to major group, because if you can see this structure, if you, the actual width of this alpha helix of protein, you can see alpha protein actually is a straight line, it is also folded into alpha helix. This width of alpha helix is around the width of this major helix, about 22 angstrom. So 20 angstrom. So that is why this alpha helix will fit into the group, major group, not in minor group. But there are very few protein that will interact with minor group also. Generally almost all protein that binds to DNA, that is called DNA binding protein. All protein interact with major group. And few protein, they also interact with minor group. Okay, did you get this? I hope you get this. Now, so DNA in our body is of B form. This B form DNA has, you can see this width. I have already told you the width is 20 angstrom and the height is about 3.4 angstrom, and total pitch contain 10.5 bases, and this I have already discussed this. The pucker is C2, Indo-pucker, and this base is positioned according to this helix. If you consider this as helix here in DNA, you can see this is DNA helix. This is helix. So this base is slightly tilted towards this helix, slightly tilted about this helix. If you draw a perpendicular line like this, so it is about 6 degree tilted. So base is tilted only 6 degree in such condition to the axis. And this is the torsion angle between ribose and base is anti. I have already explained anti and syn. So you can see this type of double-stranded helix that is present in our body. But there are other form of discovered DNA structure, and that are A form and Z form. When you dehydrate this DNA, means if you place that DNA into a water-free environment, so what will happen? This DNA A will become this type. You can see A will become like this, constricted. So it will become constricted. So you can see in that constricted base there will be about 11 base pair per pitch. So width will be about, here you can see the width is more because it is constricted like this. Dwarf. A is a dwarf DNA. So what will happen is width will increase, but length will decrease. Dwarf. Width will increase, length will decrease. And then in that condition, the distance between base pair will lessen. And then this angle with axis also increases. It becomes 20 degree. Here you will find C3 endo configuration of the sugar. You will find deoxyribose sugar is in the form of C3 endo configuration, and then glycosidic bond conformation is in NT. Now let's talk about Z. So Z is elongated in comparison to this B. So this is most dwarf. It is like dwarf. It is in the middle. It is elongated, most elongated. You can see this A form is most dwarf. B form has more height than this A form, and this is the most elongated form. So when do we get this most elongated? When you add salt to this. When you add salt in salt environment, this DNA will form a zigzag form. This Z for zigzag form. Now you can see the breadth will be shortened because height is elongated. Now one pitch contain 12 here, 12 base pair, and this base length is elongated because height is, this breadth is shortened, length is little bit elongated, 3.7, and you can see this 7 degree angle, and endo is C2 like this. Only A has different, and sometimes you will see in case of purine you will see C3 exo. So both for pyrimidine it is endo, and for purine it is C3 endo. Now NT for pyrimidine and syn for purine. So this is zigzag. That is why NT for NT, the pyrimidine is in NT and purine is in syn configuration, and that is why this will get a zigzag type of form. You can say, remember this very simple form. So this is must dwarf. A is must dwarf. B is slightly elongated, and then this Z is most elongated and zigzag. So you will see this pyrimidine in NT, pyrimidine in actually NT, and purine in C. So that is why this attain the form of zigzag form, and we actually are concerned about this B form. So if you must have to remember the B form only. So if you understand B form, you can compare this B form with A form and Z form. A form is attained by dehydration, and Z form is attained by addition of salt. So in salt or in conformation, it takes a Z form. So I will ask question from this. So in which environment does it take this B conformation will take the form of Z form? So, and then what is this configuration of this pyrimidine and purine in Z form? So what is the configuration of pyrimidine and purine in Z form? What is the configuration of this pyrimidine and purine in A form? So A form always NT, but you can see in Z form NT for pyrimidine and syn for purine. So remember this part, puckering and glycoside. So this you can see the width is more here, and width become little bit less, and width again less. So width decreases, this height increases. Height increases. Here you can also see this width is highest in A form and width decreases and then height increases. You can see the zigzag form. So, and the base pairing in this both two form, you can see there is not a regular base, this pair formation. Hydrogen bond formation is not a regular. You can see some bases are out of this double helix, and here you will feel many very less base flipping. This is called base flipping. When base come out of this double helix, it is called base flipping. This is called base flipping. This base flipping is very less in this B form. Base flipping you will see very high in Z form and A form. So this you can compare this. This is A form, Z form, B form. So these are the picture. Now let's talk about the organization of DNA. So once DNA has, we have already studied DNA has attained double-stranded helical structure. This double-stranded helical structure. Okay. This is double-stranded helical structure. Now this double-stranded helical structure will fold. This is double-stranded helical structure. I have already told you the length is 20 angstrom. And this 20 angstrom means 2 nanometer will fold into this form, 10 nanometer. And this DNA actually, this strand, consider this as this strand, okay, like this. So this is 2 nanometer. This will fold here on some bead. We will talk about this bead later. So it will fold like this. This is a bead, okay. So this DNA will fold over this bead like this, okay. Then other bead come here like this. So this DNA again will fold over this bead. Like this. Again here next bead comes. So this DNA folds over this bead. So this is the first level of folding. This is called bead on string structure. This is called beads on string structure. This is called beads on string structure. So a string is folded on beads. So you can see 2 is converted into 10. 2, 10, 30. So T for 2, T for 10, T for 30. So this beaded structure again will fold like this, like helical, in a helical sense. So this is a 30 nanometer chromatin. This is called chromatin fibril. This is bead on string structure. This is called chromatin fibril. Bead on string structure and chromatin fibril. Now this chromatin fibril, this fibril will fold on a different type of scaffold. What does a scaffold means? Fold like this. And this structure you can see, this structure will fold on a scaffold. What is a scaffold? Is a platform protein. Okay, it is a platform. It will fold on some platform. This whole structure, this structure. This is represented by this. If this straight line is represented by this 30 nanometer chromatin. This is chromatin. So this is chromatin fibril. Remember the name terminology. Fibril. This is chromatin fibril. This is bead on string structure. This is bead on string. This is CF, chromatin fibril. This chromatin fibril again fold on some protein. This is bead or histone. This bead protein are called histone protein, and this non-histone protein, this is other type of protein. It will fold like this on other protein. Okay. Now this folded, you consider this folded like this. So this folded form again will again refold in circular fashion. So it's difficult to imagine, but remember this protein, it will fold like this. Like this. So this beaded structure, this condensed 300 nanometer again will condense into 700 nanometer, and this 700 nanometer is nothing but chromatid.
Oh, okay.
So this is how actually the whole DNA, if you join all the DNA of our nucleus, the whole DNA will be about 6 feet. Its whole DNA is about 6 feet, and this 6 feet DNA is compacted in this form. First stage, second stage, this is third stage, and this is fourth stage. In 4 stages, this DNA is compacted in a very tiny form that can be arranged on a tip of a what do you say, nail, tip of a nail. So it is like it will arrange in a tip of nail, 6 feet long, that is 2 meter, about 2 meter long DNA. Whole DNA will compact in a very tiny nucleus that is not visible from your eyes. So this is the DNA organized. This is how DNA is organized. Now question comes in our cell. Okay. So you understood first this double stranded DNA. So remember double stranded DNA, double stranded. Then second is boss, B downstream, and third is CF. And then this CF will fold into this chromatin scaffold. It will fold into this 300 nanometer structure, and this 300 into 700 nanometer structure. So this 700 nanometer structure is chromatid. So this is how it will. So you need to remember this boss, B downstream structure, and chromatid. So our body, in which form right now, our body when in nucleus, our nucleus, what happens? This DNA will produce RNA in permanent cell. DNA will produce RNA, and RNA will produce protein. This mRNA will produce protein. So in what form does this DNA exist in our body? In our body, in permanent cells, this DNA are generally, the DNA exists in this chromatin fibril. That is why this is called chromatin fibril. Okay, 30 nanometer form. Did you understand this?
Yes, sir.
Which gene, the gene which are in on condition, which gene are in switched on? Switched on gene are in these two conformation, not in this conformation. Switched on gene. So this conformation will be attained before the division of cell. This conformation will be attained before division of cell. So our cell has this type of conformation, especially switched on gene, those genes which are constantly producing RNA and DNA. You already know, do you know one fact, this some genes in our cell are switched off and some genes are switched on? A set of gene is switched on, is on in liver. The same set of gene is not switched on in muscle. That is why muscle is different from liver. Gene is not switched on. You know, our genome, whole genome has about 30,000 genes. 30,000 genes. And all genes are not expressed in all tissues. Only maybe, let's say, 10,000, one set of 10,000 is expressed in liver. Other set of 10,000, maybe 9,000 is expressed in muscle. And that is how our tissue actually are different from each other. And those which genes are switched on, they are in this condition. Those chromosomes are in chromatin fibril or bead strand structure. So let's see this. What is this bead? So you can see this. You can see it from this. This is DNA. DNA is folded like this on this bead strand, and then bead strand again will recoil. There will be more recoiling, and this recoiling will form this chromatid. And this chromatid is actually, chromosome is actually seen during the metaphase condition. This is metaphase. Chromosome is a metaphase genetic material, metaphase, genetic material. So you have to understand. So this is how it will fold. So this B downstream structure, and this is chromatin fiber like this, and then it will further condensation, two-step condensation will form chromatin. Understand this condensation. Now, what is this bead? Bead is very important. This bead is histone. This bead is histone. So, this histone is an octamer. Remember, it is an octamer. Octamer made up of 4 tetramer. This 4 and again 4 tetramer. These 2 4 tetramer, when they come on each other, this becomes octamer. So histone is an octameric protein. That means it has got 8 protein. Now this octameric protein is made up of this H2, H3, and H4. H2 is H2A, and then H2B. So this H2A, H2B, and this H3 and H4, they will come together and they will form an octamer. You need to understand this much only. Did you understand this? What are the proteins? Histone proteins? H2A, H2B, H3 and H4. So this will form octamer. These 4 protein will form octamer. And this octamer is nothing but this octamer is bead. And these all histone protein, H1, H2 have a terminal, a protein, free protein. You can see this free protein. This free protein come out of this bead. And this free protein are called histone tail. They are called, you can see, tails and terminal tails of histone. These tails are very important for gene expression. Gene expression. These tails are very important for gene expression. So making gene on and off. That is why these tails are important. All of these 4 histone have got this tail. So here is tail. Though it is not shown here, here is tail. So this all has tail. You can see this tail. Tail part is there. So these are histone. They will form like this. Now we will talk about from this in next class.